biology seminar for the invitation. And also everyone who's here for attending my talk. And this research wouldn't be possible without the collaboration with a lot of other people. And in particular, I have colleagues at the Geophysical Fluid Dynamics Lab, which is affiliated with Princeton University. I also have some colleagues at the University of California at Irvine and at the Technical University of Denmark. And this work further wouldn't be possible without the generous support of several different funding agencies. So I have basically three goals for this talk. And I'm primarily a biogeochemist. And biogeochemistry is a field which, I guess, integrates different elements of biology, geology, and chemistry to explain the dynamics of different elements and chemicals in ecosystems and the environment. And that field is a bit of a mouthful. It's a bit of a complex field, and I wouldn't be surprised if some of you have never heard of it, because when I started working working in this area, I actually had never heard of biogeochemistry. So I'm going to try to do my best to give a suitable introduction to the basic concepts of biogeochemistry, and hopefully maybe convince some of you that it's an interesting and beautiful area, despite the fact that it involves so much so much grunge and messiness. And then I'm going to pivot a little bit and talk about how many of the models which are used to make predictions or understand phenomena in global biogeochemistry, which are based on traditional ecosystem models at their core, have some inadequacies. And I will try to tie these inadequacies to a lack of representation of biodiversity. And in doing so, I'll try to motivate and introduce a different modeling framework called trait-based modeling, which has two primary advantages. It provides a really efficient way of encoding a tremendous amount of biodiversity, and it does so without introducing, an explosion of unknown or unconstrained parameters. And the types of models that arise from trait-based modeling appear a little bit different in form than the kind of classical models you might see in a mathematical biology textbook. And I hope to convince some of you that this trait-based modeling is potentially an interesting area for mathematical biologists to investigate. And then the very last thing I'm going to do is I'm going to apply trait-based modeling to a very specific example in ocean biogeochemistry. And I'm going to show how we can use it to improve the fidelity of ocean models, or improve their qualitative and quantitative performance. And in particular, I'm going to show that we can use traits to predict how the elemental composition of phytoplankton varies. So the carbon to nitrogen to phosphorus ratio, so the elements that compose something called the marine phytoplankton, how that can be predicted, and how including that in models can lead to the removal of some pretty persistent pathologies. So most of the results, or most of the stuff I talk about in this talk, is contained in these two papers. So the first one, the one on the right, is kind of a review/motivation paper that talks a lot about trait-based modeling and its importance, or its utility as a tool to predict emergent phenomena in really diverse ecosystems. So that's one of the parts of this paper on the right. And then most of the results on biogeochemistry are in this preprint on the left, which is on the Earth System Science Open Archive as of maybe a few weeks ago, or a week ago. Okay, so let's get started. So one of the most important phenomena in ocean sciences, in marine biogeochemistry, is something called the biological carbon pump. And here I have a figure, which is a cartoon that illustrates the process. And one of the key players in this carbon pump are marine phytoplankton. And marine phytoplankton are single-celled organisms which live in the surface of the ocean and perform photosynthesis. So they basically are engines that absorb carbon dioxide, they absorb light, and they absorb inorganic nutrients. So molecules like phosphate or nitrate or ammonium, which aren't bound to carbon, and they combine those elements into organic matter. And then that organic matter undergoes a further series of transformations. So zooplankton are the predators of phytoplankton. So they will eat the phytoplankton, and then they will be in turn eaten by larger predators, and they will release like particles which contain a lot of organic carbon, and those particles will be decomposed by heterotrophic bacteria, returning the carbon dioxide and converting some of the organic nutrients into inorganic nutrients. And so there's actually a cycle of carbon and nutrients involved in this process. But as a result of this cycling, some fraction of the organic carbon that phytoplankton fix is transported deep below the ocean surface and remains there for a considerable period of time, many thousands of years. And that has a huge environmental impact. And so sadly, I have to keep changing the factor every time I give this talk because we release so much CO2 into the atmosphere. But if we stopped this biological carbon pump, the atmospheric CO2 in the ocean would rise by a significant factor, I think a factor of 2 nowadays. So this is something that's very important to understand. And the ultimate rate of this process is very strongly controlled by physical processes in the ocean, like ocean mixing, which supply inorganic nutrients to phytoplankton. And those inorganic nutrients you can kind of think of as analogous to the fertilizer that we put into crops. So it's also important to get a basic idea of what marine ecosystems in the surface are like. And so, I'm sorry. So here is a satellite image which contains estimates of annually averaged surface chlorophyll concentrations in the ocean. And this is on a logarithmic scale. You can't like exactly say this is proportional to biomass, but you can see that there's very strong gradients in marine ecosystems. And these gradients are reflective of the broad scale features of the ocean circulation. So there are basically what we call gyres at the ocean surface. And so there's a clockwise, is that right? Yes, a clockwise circulation in the subtropics in the northern hemisphere and a counterclockwise one in the southern hemisphere. And that actually suppresses the supply of nutrients from the deep ocean. And so you get you get these large ocean deserts in the subtropics. And then you have highly productive regions towards the poles and at the equator, where the ocean circulation tends to be in the opposite sense. And we can use that to define 3 ocean biomes, which I'll call the oligotrophic gyres, the tropics, and the subpolar gyres. There's of course more complex and higher resolution ways to define biomes, but for our purposes, this classification will do. So you might wonder how you would go about modeling the strength of the biological carbon pump. And to do that, to understand how to do that, we need to introduce a concept called ecological stoichiometry. The idea of ecological stoichiometry is to try to represent life as a chemical reaction. And here I've written out a kind of, not sure what the right word is, a kind of formal version of a chemical reaction that represents the fixation of carbon, nitrogen, and phosphorus into the organic matter of a phytoplankton. And these ratios of organic matter, so 106 carbon to 16 nitrogen to 1 phosphorus, roughly characterize the mean, or the average composition of marine phytoplankton throughout the ocean. And if you were to assume that that you could characterize all phytoplankton by this ratio, then it actually would be relatively straightforward to come up with a low order estimate of the strength of something like the carbon pump just by mass balance considerations. So it turns out that in the surface of the ocean, the concentrations of inorganic forms of nitrogen and phosphorus are very close to zero. But in the deep parts of the ocean, those concentrations are pretty high. And so that means that there's an upward flux of inorganic nutrients from the depths to the surface due to ocean circulation. And so therefore, by mass balance, there has to be an equal flux of nitrogen and phosphorus in some form downwards. And it turns out that that form is the export of this, the export of organic matter. So basically, this type of molecule traveling downwards. And so you can come up with a quick estimate of how much export there is in a given region just by multiplying the C to P ratio of organic matter times the supply of inorganic phosphorus from the deep ocean. And so one quick caveat, there's nitrogen and phosphorus are two of the most important nutrients that phytoplankton use to survive. An additional important nutrient is iron that won't be an important subject in this talk. But nitrogen and phosphorus have a bit of a different dynamic in the ocean. So it turns out that nitrogen is by far the scarcest resource for phytoplankton. So if you were to, go to a random part of the ocean, take a bottle of water and dump nitrogen into that bottle, dump inorganic nitrogen into that bottle, you'd see a little mini bloom of phytoplankton. So we say that nitrogen limits the growth of phytoplankton in the majority of the surface ocean. However, nitrogen isn't really a conserved tracer like phosphorus is. And that's because there's this really large pool of dinitrogen gas in the atmosphere, and it's also dissolved in the ocean water. And phytoplankton and actually heterotrophic bacteria are able to access that pool, even though it's energetically costly. And so it turns out there's a process called nitrogen fixation, which kind of adds just enough nitrogen to the system so that phytoplankton are limited by nitrogen, but kind of on the edge of being limited by phosphorus. But because of that reason, you sort of have people tend to focus on phosphorus and carbon to phosphorus ratios in these mass balances. All right, so understanding this ecological stoichiometry is really key in biogeochemistry because it basically sets the strengths of the coupling of the cycles of all the different elements. But typically in biogeochemistry textbooks and in all the major ocean models, the potential variation of these ratios away from their mean value is ignored. And one of the main topics of this talk is going to be investigating the causes and consequences of deviations of this ratio from the mean. But before we get to why the ratio might deviate, or if it even does, I want to do a quick detour into talking about how one actually models these things in practice, and some of the shortcomings of typical Earth system models. And so here I have a picture on the right of the Geophysical Fluid Dynamics Lab's Cobalt Model, which is their global biochemical model. And you can see that it looks highly complex. It represents many different pools of matter, including small phytoplankton, large phytoplankton, diazotrophs, a variety of different zooplanktons, which engage in some kind of food web, pools of detritus, pools of different types of dissolved organic matter. And so you end up having this really big system of basically like ODEs that are being advected around with a tremendously large amount of parameters. So I can show you this is just a small subset of the equations involved in the nitrogen dynamics. And the highlighted box or the phytoplankton portion of that, which is what I'm going to be messing with and modifying. But despite having despite this model being so tremendously complex, it's actually only representing a very low amount of biodiversity. And what do I mean by that? So if we were to kind of zero in on like this first term in these equations for the change in the nitrogen bound in phytoplankton, we would see J prod of small phytoplankton and large phytoplankton and diazotrophs. And the strength of that term would be based on the growth rate of those phytoplankton, which would be modeled by an equation that looks like this, where the growth rate is a function of the concentrations of different resources in the environment. So it's a function of the amount of phosphorus, the amount of phosphate, the amount of nitrate and ammonium, and the amount of irradiant. So these are all really important nutrients for phytoplankton, or important environmental drivers for phytoplankton. And the functional response encoded in this equation basically is one that's derived from studying a single organism in like a laboratory environment. So if you take a single phytoplankton and you grow it in the lab, you vary the conditions of these resources, you'll get some functional response that initially is linear in the concentration of resources and then saturates at some maximum value. And basically, these ocean ecosystem models pick like one or two such functional response curves and use them to characterize like the entire plankton ecosystem, which in actual fact contains I don't know, really an mathematicians, really like a functionally infinite number of different species. And this leads to some negative consequences. And in particular, if you try to use an Earth systems model to predict the functional response, let's say, of the entire ecosystem to environmental drivers. And so what do I mean by that? So something like how the growth rate of phytoplankton vary across the ocean as a function of temperature, or how nutrient uptake rates vary across ocean ecosystems where nutrient concentrations exhibit a gradient, or for zooplankton, how zooplankton grazing rates depend on the concentrations of their phytoplankton prey. So those are sort of like different examples of ecosystem level functional responses. And in these ecosystem models, the predicted functional responses tend to retain the flavor of the one or two hyperbolic functions that you use to parameterize the phytoplankton in the model. But when we look at real ecosystems, we tend to see a much smoother and broader functional response. And what we believe is happening is that in reality, so here I've drawn like two functional responses that characterize different types of phytoplankton and show different types of trade-offs they might exhibit. And so this is growth rate as a function of some resource concentration. In actual fact, there are an entire family of these curves. And within the ocean, the ecosystem, there's a kind of competition between organisms that have a lot of different possible curves. And in a given location with some set of environmental drivers, one or a small subset of those organisms with a small subset of those curves win, but it's actually different everywhere in the ocean. And so you might end up seeing, so what we might actually be seeing in real ecosystems is more of a curve like the black one, which reflects some aspect of all the different functional responses that are possible in phytoplankton. And so this lack of diversity, we believe, leads to mismatches between observations of ecosystem processes and predictions of what they should be based on models. And there's an additional issue here, which is that if we were to imagine trying to use an Earth systems model to predict what will happen either in a future ocean or in a past ocean, which is a really common use of these models, we might be concerned that the out of sample prediction could be bad, because the Earth system models have a ton of parameters, and those parameters are kind of informally tuned to recapitulate modern ocean conditions. But in a future ocean, we might end up with completely different species of plankton dominating in different areas, and so we might expect the performance of Earth systems models to degrade further outside their tuning envelope. And so here I'm going to be focused. Sorry, I just realized I did not plug in my laptop. And so my laptop is about to die unless I make sure it's plugged in. Okay, well, not sure if I've done it right. We'll see. Maybe it'll be okay. So here I'm going to be the functional response, or the ecosystem level functional response that I'm going to be most interested in is phytoplankton stoichiometry. And I told you earlier that most models assume that it's a constant, either a global constant, or maybe if a model has a few different phytoplankton types represented, they might assume that the ratios of elements of those phytoplankton types are vary between the different types, but are constants. In the real ocean, we see some sort of variability, and the variability is actually systematic, where in high latitude ecosystems and near the tropics, we tend to have lower carbon and nitrogen to phosphorus ratios. And in subtropical ecosystems, we tend to have higher nitrogen to phosphorus ratio. So it seems like in the ocean deserts, we have very efficient use of nutrients and very efficient export, whereas in the productive tropical and subpolar ecosystems, we have somewhat less efficient use of resources and somewhat less efficient export. And you can also take a look at how carbon to phosphorus ratios vary according to environmental drivers, and you can see that there's some clear patterns. In cold waters, you tend to have lower carbon to phosphorus and nitrogen to phosphorus ratios than in warm waters, and similarly, under low nutrient conditions, you tend to have very high carbon and nitrogen to phosphorus ratios compared to under high nutrient conditions. And if mechanisms that cause these patterns are operating in the ocean, they could introduce new and interesting feedbacks into the Earth system. So it might be potentially important to capture these. And in particular, many Earth systems models exhibit a certain set of pathologies, which we have reason to believe are related to the use of static stoichiometry formulations. And these pathologies are basically tied to the representations of the phosphorus and nitrogen cycles. And so in particular here, I'm showing two plots of simulations from the Cobalt model, which I showed on a few slides ago, with static stoichiometry. On the top plot, I'm showing which nutrient is limiting phytoplankton. And so the blue, the really light blue and medium blue indicate varying stages of phosphorus limitation. And you can see that there's a really enormous phosphorus limited region in the Atlantic Ocean, and also there's also a large area of phosphorus limitation in the Indian Ocean, and even some off the coast of Japan. And these phosphorus limited regions really aren't seen in the real ocean. So this is a bit anomalous. And a consequence of this pattern of nutrient limitation is that you get suppression of nitrogen fixation in regions where phosphorus, not nitrogen, is limiting. So as I said, nitrogen fixation is a costly process. And so if nitrogen is abundant and available to phytoplankton, then there's no niche for organisms that might fix nitrogen. And so the models tend to predict an absence of nitrogen fixation from the Atlantic, which is much lower than observations. And even worse, the nitrogen cycles in Earth systems models, in particular in GFDL's Cobalt model, don't come to an expected balance. So simulations, even over really long periods of time, are losing nitrogen, and the overall rates of nitrogen fixation are much lower than expected. And there are a few other pathologies relating to patterns of carbon export, which tend to be underpredicted in ocean gyres. So I'm going to pause for 1 second and actually see what's wrong with my charger. I'm sorry, my battery, my laptop is plugged in, but somehow my computer is saying that it's losing charge.
I know that all too well, actually.
Oh wait, okay, I fixed it. I just had to replug in. I just had to unplug the adapter and plug in. Okay, so I'll continue. Okay, so what should we do about this? So I argue we should take a different approach to how we build ocean ecosystem models, or other models where diversity is potentially an important factor. And so the approach that I'm going to argue for is called the trait-based approach. And the basic idea is that instead of defining the compartments in your model using several characteristic species, which you think are important, instead you should define the organisms in your model using their traits. And so what are traits? So traits are things like the radius of the organisms in your model, or the types of nutrients that they are able to use, or how they allocate their resources between different physiological functions. And if you formulate a model in this way, you end up with a trait, like your sort of lowest level model will involve an equation for the evolution of something called a trait distribution function. So I have a very simplified equation of what that might look like here. So here, theta is a parameter defining a point in trait space. E is some environmental driver, and then you have to derive some growth rate as a function of trait. So you have to derive a functional response for the organism with a certain given combination of traits theta. But if you do that, then you can, at least in principle, formulate a model for the evolution of this trait distribution function. And so why is this approach any different or better? Well, one of the advantages of this approach is that by switching to a view that's focused on traits, you're often able to use trade-offs between the traits and bring in first principles from things like biophysics or physiology or biochemistry to strongly constrain the functional responses that are possible in a cell. So a really good example of this is the size of a cell. The size of a phytoplankton cell determines almost everything about how that organism can perform. So it determines, if you know fluid mechanics, like the diffusion equation of with a cell that onto a sphere that's absorbing like some sort of compound. That's the rate of diffusion, the rate of flux to the sphere is really strongly dependent on radius, and that has implications for the ability of a cell to uptake organic matter. Things like the types of predators that can consume you, that eat you, like those, that's very size dependent. Something like the self-shading of chlorophyll, size dependent too. But there's generally speaking, there's more than that. There's like a lot of other different traits, and trait-based ecologists have been working to figure out like how organisms with different trait combinations are constrained. And oftentimes, if you adopt this trait-based framework, even though you have a much more complicated equation for an entire trait distribution function, it ends up having much fewer free parameters because the functional responses are generally like constrained from first principles. So you end up having a model which functionally encodes a continuum of possible species, right, as defined by points in your trait space. And it does so having actually few free parameters because it's really strongly based on first principles. But you have this other problem, which is that now you, instead of having an equation like an ordinary differential equation for a few different species, you have an equation for this trait distribution function, which is a lot more costly to store and a lot more complex. And so the trade-off is you need to make some kind of approximation to those dynamics in order to solve them. And there's a few different possible choices of dynamics. So one of them is called a system of infinite diversity. So here I've drawn a little maquette of an NPZ model where there's some nutrient which is consumed by phytoplankton, and then the phytoplankton are consumed by zooplankton. And so normally there would be one P in this model, but let's say you took a trait-based approach to phytoplankton, there might be some important trait, and you could define a continuum of different phytoplankton types, and you could just discretize that distribution and solve it via brute force. So this is the most crude way of handling a trait-based model. So the lightest weight way of handling a trait-based model is something called an optimality approach. And this only works in certain situations where you can actually solve for an optimum trait combination. But here the idea is that you assume that the dynamics of selection or of competition between these phytoplankton plays out quickly, and that you can characterize the winning phytoplankton type. Let's say that all these phytoplankton types have different functional responses, different curves characterizing them. You can, as a function of resource concentration, you could say that perhaps the ecosystem as a whole is going to be characterized by the single phytoplankton type with the fastest growing trait combination. And so basically you will replace your hyperbolic functional response with this like optimized envelope functional response, which characterizes all the different curves that take place or that are possible within your model. If you want to be, and so this is actually the approach that I'm going to take in my modeling. You can do more sophisticated things depending on what your situation calls for. You could also start writing down equations for the moments of the trait distribution functions, such as its mean and its variance, and you start to get something that looks a little bit like fluid mechanics, where you're deriving closures of these of this hierarchy of moment equations. You can also try to use adaptive dynamics to figure out if there's like some sort of evolutionarily stable strategies or some way to predict which trait combinations will arise in certain areas. And lastly, if you have a very high dimensional trait space, so many different traits, where maybe solving for an optimum doesn't make sense and a system of infinite diversity is impossible, you can actually just randomly sample. So you can kind of take a Monte Carlo approach and just seed your system with like 100 samples from your trait space and just look and see what emerges. And that actually lets you reproduce the emergent biogeography of phytoplankton with very little external assumptions. Okay, so I said I was going to take an optimality approach, and I was going to try to use traits to predict phytoplankton stoichiometry. So how am I going to do that? So here is a picture of a phytoplankton cell, or a little drawing of a phytoplankton cell, and I'm interested in how much carbon, nitrogen, and phosphorus it's going to have. And so I could say, well, the that's going to be determined by how this phytoplankton allocates its resources between different biochemical functions. And so it turns out that the primary phosphorus-containing molecule in most living things is the ribosome, or is RNA, but RNA is a component of ribosomes. And if you don't know, ribosomes are I don't know if I could call them an organelle. They're like a unit that's responsible for protein synthesis. And pretty much every molecule in the cell contains a decent amount of carbon and protein. So the ratio of ribosomes to other parts of the cell plays a really big role in determining what the carbon and nitrogen to phosphorus ratios of the cell are. So it turns out that there are a few other pools of phosphorus. So phosphorus can, and some of them we don't quite understand as well the physiological role that they play. So there's compounds that seem like luxury storage compounds, like polyphosphates. There are phospholipids, which exist in the membranes and which can be substituted for non-P containing compounds. But the basic idea is we're going to try to define the traits of this organism in such a way that enables us to predict the balance between ribosomes and proteins, and then perhaps do something for these other compounds too. And so if you do that, you naturally get three different hypotheses for three different mechanistic hypotheses for how environmental factors might control stoichiometry. And so the first is something called the growth rate hypothesis, which basically is the idea that, well, a cell needs a lot of ribosomes if it's growing fast, because protein synthesis rates scale with the biomass of ribosomes in a cell. That's a pretty well-tested empirical fact. And so you might expect, if a cell was in oligotrophic conditions where there were few resources and it was growing slowly, it would also have few ribosomes, and thus it would have a high N and C to P ratio. Whereas if you had a ton of nutrients and cells are growing very quickly, the cells would require a lot of ribosomes. And as a result, they would have a high ribosome to protein ratio and a low carbon to phosphorus and nitrogen to phosphorus ratio. And this is consistent with the data that I showed you earlier. So there's an interesting corollary of the growth rate hypothesis, which is called the translation compensation hypothesis. And it's based on the idea that different physiological processes within phytoplankton may have different dependencies on temperature, just because they're different chemically. And in particular, it's well known from biochemistry that almost every protein catalyzed or ribosome catalyzed process increases in speed with increasing temperatures. And the rate of increase has been pretty well characterized of those processes. On the other hand, for phytoplankton, there are a lot of photosynthetic pigments. And these photosynthetic pigments, they capture photons and they transport electrons. And those processes turn out to be very quantum mechanical in nature. And actually, if you study them in a certain way, they seem to exhibit very little temperature dependence. And so there's this hypothesis that the light harvesting apparatus of phytoplankton, so phytoplankton basically have to build chloroplasts or chlorophyll in order to get carbon, that the kinetics of that of the investment in that pool may not vary very much as temperature changes. And if that was the case, in warm waters, you would expect there to be a much bigger investment in the very N-rich light harvesting apparatus, and a consequently smaller investment in ribosomes, and thus a higher N to P ratio in warm waters. And conversely, in cold waters, the ribosomes get a lot slower. The light harvesting apparatus doesn't get any slower, and so you might expect there to be more ribosomes in cold waters, and thus you would see higher N to P ratios in warm waters, which is also consistent with the data that I showed you. And then the last I guess, hypothesis for how stoichiometry might be regulated mechanistically is something called frugality. And the idea here is really simple. If a resource is scarce, a cell might find a way to use less of it. And if a resource is abundant, a cell might find a way to use more of it. And then the plasticity of a cell quota then might determine how much a cell responds to different conditions. And so it turns out that cells aren't very able to change their carbon and nitrogen quotas because they already have a lot of both elements, and pretty much every molecule they have contains carbon and nitrogen. P, on the other hand, is a lot scarcer in cells. It's only contained in a subset of molecules, and there are some compounds which contain a lot of P, but we don't quite know. We're not quite able to like mechanistically determine what role they play in the organism's survival. And so you could say that perhaps just the concentration of phosphate might control stoichiometry, so that when phosphate is scarce, organisms find a way to use less phosphorus and have a high N to P ratio, and consequently when conversely, when phosphate is high, they do the opposite. They find a way to use more phosphorus. Maybe they put it into luxury storage pools and they get a lower C to P ratio. So we developed a trait-based model which encodes all of those hypotheses kind of in one framework. And we basically represented a phytoplankton. We defined a phytoplankton according to its values of these 4 traits. And so these traits are the cell radius. So that's R. A is an allocation of proteins devoted to nutrient uptake, specifically to phosphorus. E is an allocation of biomass to biosynthetic proteins and to ribosomes. So this is the pool that's very rich in phosphorus. So this pool E. So when E is high, the C to P ratio is low, and vice versa. When it's low, the C to P ratio is high. And then L is the level of photosynthetic proteins, which are used to harvest light from the environment. And then there's also a storage pool, which is a place where we'll have to have some kind of phenomenological parameter that characterizes the rate of storage. And so from if we define a cell in this manner using these traits, we can actually derive a series of equations for the growth rate of a cell with a given trait combination in an environment defined by concentrations of nitrogen, phosphate, level of irradiance, and temperature. And we can do that using basic like laws of physics. So diffusion limited uptake rate divided of nitrate divided by the quota of the cell for nitrate, which is determined by the level of the traits, for example. And we can do the same. We can do the same for phosphate. Or there's a little extra complexity in the expression for phosphate uptake. I won't get into the details of that. And then we can put in different temperature dependencies of these processes to characterize the difference in photosynthetic functional response and its dependence on temperature, and the kinetics of the ribosomal pool. So here there's a growth rate due to protein synthesis, and it's just a function of how much you've invested in ribosomes. This is an equation for how much carbon, basically how much carbon you're fixing, and I'm not writing it out because it actually takes a bit of time to explain it. And then we could say that the overall growth rate is the minimum of these 4 rates. So basically there's these 4 processes: nitrogen, phosphorus, carbon uptake, and then biosynthesis. And the cell has to do all of them in order to grow, and the slowest one is the one that limits the growth of the cell. And then we can say that we can then try to find the optimal strategy, which is the optimal trait combination or the trait combination which leads to the highest growth rate. And it turns out that we can, for this model class, we can do that pretty efficiently. And this encodes all the different hypotheses that I mentioned. And we actually were able to embed this model into a Bayesian computational framework and show that it quantitatively reproduced observed patterns of phytoplankton stoichiometry from some really high resolution cruise data that my collaborators collected, and that our model actually performed a lot better than some other more phenomenological models of phytoplankton stoichiometry. So basically, we created a decent model of trait-based model of phytoplankton stoichiometry, and we wanted to see what it would mean for biogeochemistry. So now I'm going to revisit the Cobalt model that I told you guys about earlier. And I said that in the previous version of the Cobalt model, these phytoplankton pools were characterized by fixed stoichiometric ratios. And so instead, what we're going to do is we're going to use the atom model, and we had to modify it a little bit to get it to work with this type of framework. There has to be a lot of compromises in ocean modeling. So what we're doing here is really like kind of a hybrid trait approach with embedded in a traditional Earth systems model. But we were able to build like a miniature atom model for each of these pools, and that predicts the optimal allocations of resources for each of a small phytoplankton, a large phytoplankton, and a diazotroph. And we performed a large number of simulations using this framework. And so what did we do? So we actually did two sets of experiments. So the first set of experiments involved comparing what we call the dynamic model to a control model that used the same functional response, but had the static ratios of the original Cobalt model. So that was one series of experiments, and so we ran basically we ran 60 years of simulations, forced by like some well-characterized past set of ocean conditions, and we looked at the last 20 years of the simulations and compared the results. And then the second set of experiments, which I don't believe I'll have time to describe, involved looking in finer detail at the different mechanisms and hypotheses I told you about, and modifying the models to either emphasize or de-emphasize those, and seeing what what the role of each mechanism was in detail. And so I'm going to, the last few minutes, I'm going to go through the comparison between the fully dynamic stoichiometry model and then the static control model. And so here's some histograms of N to P ratios. And so here I actually defined 4 biomes, so one more than the 3 I told you about in the beginning of the talk. And I have some observations of N to P ratios from data sets, and those are in green. And I have the control model in orange and the dynamic stoichiometry model in blue. And you can see that the dynamic stoichiometry model is much better at capturing the variation of stoichiometry that appears in real data sets. So there's, for example, like in oligotrophic gyres, there's a shift to much higher N to P ratios, and the static control model doesn't capture that. So the static control model is like has different ratios for large and small phytoplankton, and there's more small phytoplankton in oligotrophic gyres. So you would think it would capture part of the signal, but it's actually quite poor at doing that. And then a similar thing occurs in sort of higher chlorophyll, more productive, like coastal and tropical regions. The dynamic model is much better than the static one compared to data. In high latitudes, all the models are about equally good. And so here's a picture of a global map comparing the static and the dynamic together. And so you can see that the dynamic has a much bigger range of variability, particularly like in the subtropics. And this greater variability, and in particular increased N to P ratios, leads to a large decrease in areas of phosphorus limitation. So in our static control model, 36% of the ocean surface was phosphorus limited, whereas in the dynamic model it was only 8%. So enabling the phytoplankton to have much more flexibility in their ratios as predicted, really reduced areas of P limitation, which is a big improvement of a persistent problem in this model. And the increases in N to P ratios and the disappearance of phosphorus limitation led to big shifts in the patterns of nitrogen fixation. So we had a really big increase in Atlantic nitrogen fixation, and actually nitrogen fixation in general. And basically, like the static control model basically had 90 teragrams of nitrogen fixed per year, which is about like half the best observations. It's actually very hard to measure and estimate how much nitrogen fixation there is in the ocean, so you could take these with a bit of a grain of salt. But our dynamic model had a lot more nitrogen fixation, and that brought the nitrogen cycle, at least as far as we were able to tell, like into much closer balance, which was something we expected. And then lastly, the dynamic stoichiometry model predicted a big increase in export in low latitude ecosystems. So in the tropics and the subtropics, so a 30% increase in export in ocean gyres and a 14% increase in export in the tropics. And in particular, the increase in ocean gyres is probably a positive, is probably an improvement. But this is something that we also expected as an effect of dynamic N to P. So I'm going to go to the conclusion slide. And skip the second set of experiments because getting a little bit short on time. But I don't want to give the impression that everything in this research was something I expected, because there was actually one very unexpected result, which was that sort of translation compensation mechanism. So that's the mechanism whereby increases in temperature drive increases in carbon and nitrogen to phosphorus ratios. And from really naive modeling, you would expect this translation compensation to introduce a totally different direction in feedbacks between temperature and atmospheric CO2. So if you know, there's a really large solubility feedback where as temperature increases, atmospheric CO2 goes up. But this translation compensation we thought was a mechanism that would counteract that, and we had like a really simplistic model which just modeled ratios directly using directly using the model prediction of C to P, and not like with a dynamic ecosystem component in the background. And this predicted that like as temperature in, let's say, the tropics was increasing, we were getting drops in atmospheric CO2, which would be quite unexpected. And we actually didn't find that when we did like a full model simulation. And the reason was that for there to be translation compensation, if you were to include it in your model in a mechanistic way, then it also has a profound effect on the way that temperature affects phytoplankton growth. And that leads to phytoplankton having a much more modest response to temperature. So when translation compensation occurs, phytoplankton don't increase their growth rate much in warm waters, and so and similarly they perform kind of well in cold waters, and that actually counterbalances completely the expected changes in export from the changes in the C to P ratios, and that was a big shock to us. But yeah, so anyway, so that maybe concludes my talk. Sorry I used up all the time, but I thank you all for listening. And are there any questions?
Thank you, George. Do we have any questions?
I have a few questions. I mean, I can just start with, I mean, one of my questions was, so you have I mean, so essentially your dynamical systems don't really have bifurcation, so coexistence and stuff like that. And I was wondering if you, I mean, that's one of my questions. The other one is, in a system with a gradient of temperature, if you have also mixing due to oceanic movement, can you achieve more variability, like, and improve a little bit even the dynamical model to match the data? Is that something that you looked at, or?
So let me answer the question about bifurcations first. So the types of trait distributions you tend to get depend a lot on, I guess, the ingredients you put in your simulation and the type of organisms, that you're modeling. So you can I think you can actually get bifurcations in trait distribution. So if you were to actually, like, so in some models where you have like continuous predators, so let's say you have like a zooplankton ecosystem where the zooplankton are grazing on each other, and each zooplankton has a size feeding preference. Depending, you basically end up getting the collapse of the system into some like discrete size classes, depending on how those feeding kernels are parameterized. So there can be some like sort of unusual and complicated dynamical effects. There can also be cases where the number of species in your trophic, like the length of your trophic web, could increase or decrease depending on your environmental drivers. So let's say you have a really full description of your ecosystem. So you have a trait distribution describing like sizes from like small phytoplankton to, I don't know, fish or something like that. Then as you like increase, I think as you increase like the rate of supply of nutrients, say, you can get like a shift in the number of species, like in the number of this sort of the size of the biggest species. That may be a bifurcation. But for what I'm doing here, it's a little bit more simplistic, where we're really solving for like an optimal strategy. We're not considering predation, and it's a bit and we know that it approximately works, but we're not actually sure. We're not like 100% sure like why it's so good at getting like the kind of 0th and 1st order patterns in the ocean. Right, so presumably, there's some coexistence. There's some like spectrum of phytoplankton of different sizes that actually are coexisting in a given place, and like how broad that is set by the grazing dynamics as well as the nutrient dynamics. That makes sense.
Yeah.
And so I'm not sure if that answers your bifurcation question.
Yeah.
And then Yeah, so yeah, okay, so when it comes to environmental heterogeneity, and like stochasticity, let's say, I think the big question is like what's like the time scale at which yeah, at which organisms, at which selection happens, and at which organisms can either adapt, right? So or so yeah, so like how fast can you get like a shift from one like organism to another, or how fast can organisms that are able to change their allocations, how fast can they do that? And then also how much actual mixing is there, like how Lagrangian versus Eulerian is the right is the fluid mechanics, because you might you might get just some kind of advection of community, so there might be some shift in the fluid flow in the surface that takes cold water from one place to another, but it might kind of bring the community with it a little bit, and so basically it's very organism dependent, like how well, let's say, how well suited to temperature, how well suited the temperature traits are. And I think you can so I think for phytoplankton, this stuff takes place really fast, and you just because growth rate differences between these organisms grow so quickly, I think you get really rapid adaptation. And so I think you would need a lot of mixing to get to kind of get a mismatch, and maybe some like average trait winning. But for other organisms it's different, I strongly suspect.
Because what is the time, sorry, what is the time scale of the circulation in the ocean, let's say, between the tropical, I mean, between the very different regions of distribution?
Oh, yeah, so I would say, I mean, the answer is it depends. So maybe like I would say like the mean circulation speed is like a few meters per second, but there's a tremendous amount of variability around that, and then there can be ocean eddies that move fast, and there are certain currents that flow incredibly fast, right? So the Gulf Stream, for example, like that's going to flow like, I don't know, I feel like a factor of 10 or more faster than the circulation somewhere else. So I feel like a year-ish, if like
okay, oh,
like something like that, like I think like order of a year for like a big ocean region, maybe more. But for like for the entire ocean, it's kind of like 5 or 10,000 years, like if you include deep mixing, which isn't what you asked, but that makes sense.
thank you.
We actually ran out of time. Do you have any more question? You will die if you don't ask. All right, if not, let's thank George again. Thank you.
And thank you guys very much.
Thank you, George.
Bye, everyone.
Bye.
I'll talk to you guys later.